Okay, this is the second installment of the paper, Is the Cell Really a Machine by Daniel J Nicholson. And I think we are on section, whatever I said. We're on section 3. And I was going to try to make this bigger for the audience this time. And is that pretty readable?
Yes. Is there a way to get rid of that left-hand column?
I don't know.
Where it shows each individual page?
I do not know how.
Okay. That would just, help us get more surface area.
It's actually kind of handy.
Yeah.
So, is that pretty clear? I think I can go to full screen here.
Yeah, it's pretty large.
Yeah, okay. Yeah, it's pretty good. So anyway, I think we want to probably start reading this. I don't think I have all my highlights in on this yet.
Okay.
Take a look, and you want to go ahead and read? Or
now, at the top, that's where it starts, part 3.
Yep, the mechanistic foundation. So it is. So we are at part 3.
Yep. Okay, I'll start. The mechanicist foundations of molecular biology have not only guided our inquiries into the cell's internal organization, they have also shaped our theoretical understanding of its basic molecular components, especially proteins, as well as championing the MCC, which is the machine conception of the cell. Monod, I don't know the exact pronunciation, also declared in his Chance and Necessity that with the globular protein, we already have at the molecular level a veritable machine. In subsequent years, as it became apparent that most proteins in the cell associate with one another to form larger complexes comprised of different subunits, a new concept began to acquire currency, namely that of a molecular machine. In 1998, Alberts, then president of the National Academy of Sciences, published a brief but highly influential manifesto titled The Cell as a Collection of Protein Machines, Preparing the Next Generation of Molecular Biologists, in which he urged aspiring molecular biologists to embrace the MCC and learn to view the cell as a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of a set of large protein machines. At the end of the article, Alberts prophesied that much of the great future in biology lies in gaining a detailed understanding of the inner workings of the cell's many marvelous protein machines.
Yeah, and Bruce Alberts is the main author of Molecular Biology of the Cell, which is kind of the Bible for molecular biologists. So he's pretty important. So, and let's see, we're up to here. It is difficult to overestimate the impact that Albert's paper has on molecular biology. And I'm not sure if we want to read everything. Let's see, he talks about, let me see here. This term is now used to describe virtually any functionally specialized macromolecular complex found in the cell. So again, this paradigm it's strong with us.
Yes, it has captured our imagination and held on to it.
It has. Some have gone as far as to regard the molecular machine conception of protein complexes as one of the most important contributions that biology has made to our understanding of how the living cell works. And there's no doubt that they do work in very intricate ways, and they form very intricate complexes. So, let's see, is because it addresses in a unified way the two classical concerns of molecular biology research, namely structure and specificity. So structure would be those protein folds, and specificity would be that when you end up with a protein in the cell, it will specifically bind to a certain class of other molecules. And like if it's like a kinase, something that attaches an ATP, it will lock onto the molecule and it'll lock on to ATP, and it's almost like a machine. So it has characteristics of that. And that's what they mean by structure and specificity.
Right, and as we mentioned last week, and I guess I'll keep hammering it home every installment. The Thomas Kuhn idea of the paradigm is that you pick a picture, a mental picture, a concept, and ideas flow from it. And so the language essentially of the Industrial Revolution, machines and factories and things like that, the picture of the factories and machines of the Industrial Revolution have informed research. And as we'll see later, too, the methodologies that science employs have informed research as well. And so it has been very useful up to a point in helping scientists to define the structure and the function of the various parts of the cell.
Yep.
And so that was a good thing for science, but what we'll be finding out is that information is being left out or just kind of hand waved away under this particular model.
And in this paragraph, historically, they're talking about how we determine the structure of proteins and DNA and all of that. And we did it by crystallizing the protein. And sometimes it takes weeks to crystallize these things. But when they can get them to crystallize, and not all proteins do this, some remain liquid, they are able to use X-rays to determine the structure of the protein.
So they actually manipulate the physical nature of the cell to make it crystallize. And is this sort of like fossilizing it?
No, it's actually, it forms a crystal. If you cool it down slowly and you have a pure protein, it will actually form a crystalline structure because it packs, like any crystal, it will pack in a certain regular order. And then they use X-rays to shoot into that, and they can, because of the diffraction of the X-rays, they can determine the structure of the protein. So they can see the helices, and they're called beta sheets. They're like big sheets that flow around. And then there's loops between them and stuff. And we can show some pictures of the cell or of proteins to explain some of that. So, but anyway, the thing is that in the cell, it never is crystallized. Okay. So we're already looking at a laboratory version of the protein. We're not looking at the wild protein, the protein in real life.
Right. The protein in the wild.
Yeah. And then the Informational School of Molecular Biology, led by the so-called phage group of Delbruck, Loria, and Hershey, used bacteriophages as model systems to investigate the molecular basis of heredity. And they came up with some of this stuff. And that was like of course, then there's Watson and Crick, who did that with DNA. That's how they discovered the helical structure of DNA, is by crystallizing it, forming these crystals. And I think he elaborates on that, and I'm not sure how much. Let's see, conceiving of protein complexes as molecular machine draws our attention to their structure. When a mechanic or an engineer studies a machine, they examine its structure carefully because they know that this will enable them to understand its operation. So function is a direct consequence of the structure, and so they elucidate the former by scrutinizing the latter. And I think they talk a little bit more about, let's see, many superlative structure. If you want to read any part of this, or you have highlighted parts on your paper.
No, I would just take notes onto a Word document if I had something I wanted to say, but I didn't really do a lot of that with this particular section, unfortunately.
Yeah. There's a part here: we must foremost know the structure of the static molecular machine at the atomic level as a precondition of making sense of its behavior and going beyond a mere phenomenological description. Let's see here. So again, I think in addition to emphasizing its structure, viewing a protein complex as a molecular machine serves to highlight the specificity of its operation. And that is how it latches on to specific parts of the DNA or some other protein or the cell membrane or whatever. So I mean, it's not to be discounted, the structural idea. But okay, so probably start reading right here. You want to start reading at despite?
Sure. Despite the popularity of the molecular machine concept, Recent research is casting serious doubts on the theoretical adequacy of this notion. Some of its problems pertain to its undue emphasis on structure, and others pertain to its undue emphasis on specificity. I shall examine each of these in turn. With regards to structure, it has become apparent that the widespread use of X-ray crystallography has biased our view of proteins. In the last 50 years, crystallographers have deduced that the tertiary structure of a very large number of proteins by purifying them into homogeneous solid-state crystals and then examining the ordered array of their atoms. So that's exactly what you said.
Yep.
Although the inferred structural reconstructions are of extremely high resolution, they represent only snapshots of incapacitated proteins frozen in time. Still, we have relied on this technique so much and for so long that it has come to shape the way we think about protein structure and its relation to protein function. Specifically, it has led to the view that each protein has a unique three-dimensional conformation corresponding to its most thermodynamically stable configuration that it must adopt in order to carry out its intended function. This deeply entrenched assumption has been called the central dogma of structural biology. So this will probably be on the exams. Were we taking a biology class? And
Right.
as I put it above, it reflects precisely how we think about the relationship between structure and function in a machine.
Yep. And the problem, of course, is that the proteins do not naturally exist in crystallized form. In fact, their native environments behave more like liquids than like solids. Proteins are really dense liquids or melted solids, consisting of a near solid exterior and a full liquid exterior. And that's important because, well, we'll let the paper talk about it. In this context, the investigation of protein structure using NMR spectroscopy. God damn it.
I know some of these words, they just kind of stick in your throat.
Well, you know, what is it, Sunday evening? It's late.
Right.
You know.
And it didn't help that I had a cup of ice cream just before doing this. That's really bad for speaking out loud.
Yeah, and I'm drinking thick coffee, which probes proteins as they twist and turn in solution, has proved more revealing. Although this technique is almost as old as X-ray crystallography, for decades its applicability was severely limited. Only recently has it become possible to employ it in large-scale studies of protein structure determination. What we have found by introducing the temporal dimension into our study of protein structure is that these proteins are highly dynamic entities that display very high degrees of flexibility, ranging from simple side chain rotations to complete rearrangements of their secondary structures. In other words, one protein can have more than one secondary or even tertiary structure, I believe. The structure of a protein is soft and fluid, not hard and rigid like that of a machine. Which means that
I think about taffy when I hear about that.
Yeah. I like the idea of the liquid exterior, you know, where it's kind of floating around, and then at the core, where where more of those hydrogen bonds form, it's more of a solid or semi-liquid, but it's capable of entering into reactions that we hadn't counted on, or one protein can enter into more than one reaction. It can have a structural specificity that is varied. You know, it can do more than one thing, and we hadn't counted on that. And it's becoming important in some of our medical research that we understand that and accept it, that sort of thing. Okay, I'll let you go.
Another important discovery prompted by the introduction of single molecule methods is that proteins in vivo seldom exhibit a single ordered conformation. What is commonly referred to as the conformation of a protein actually comprises a range of well-defined configurations separated by low-energy barriers that a protein molecule continuously samples by means of stochastic fluctuations. Any population of seemingly identical proteins is really a heterogeneous mixture of molecules with slightly different conformations in equilibrium. It is just that Classical structure determination methods identify only the predominant conformation in the population, averaging out the differences that exist between the individual molecules, a problem that I shall discuss in more detail later on. This difficulty is compounded when faced with proteins that do not have a predominant conformation. For example, the globular protein lymphotactin, okay, got it in 2 tries, that's pretty good, adopts 2 completely distinct alternative conformations, and it undergoes major structural changes as it flickers from one to the other. More generally, it is important to realize that the conformational landscape of a protein is not fixed. The binding of ligands, post-translational modifications, temperature, pressure, and solvent concentration can all alter a protein's conformational landscape by changing the height of the energy barriers that separate its alternative conformational states.
Okay. Lastly, and perhaps most surprisingly, many proteins do not have an ordered conformation at all, but instead roam the cell as unfolded polypeptide chains. That would be, the creationists would say that was another big surprise. This major class of proteins, which have only recently come to the attention of researchers, are called intrinsically disordered proteins. And these are kind of interesting, so that is a nice thing to highlight. And they are found in all domains of life. In mammals, it has been estimated that approximately 75% of the signaling proteins and about 50% of all proteins contain at least one disordered region of more than 30 amino acids, and as many as 25% of all proteins are completely disordered, that they never fold. So they're just knocking about in the cell in their unfolded chain-like form until they do something. It is possible to make such calculations because IDPs differ from ordered proteins already at the level of their amino acid sequence. Peculiarities in this sequence define both the ability of ordered proteins to fold and the ability of IDPs to stay unfolded. Well, the former have funnel-like conformational landscapes with a well-defined energy minimum that refers to the landscapes they do of proteins where they show that the preferred folded states have like a low energy dip. And I can't visualize the funnel-like conformational landscapes, I guess, would be where you dip into this huge funnel like a whirlpool. And it tends to make it happier when it's at that lower energy state. Well, the former have funnel-like conformational landscapes with a well-defined energy minimum. The latter have flatter conformational landscapes, okay, with numerous local energy minima. So the ones that would fold into a stable state would hit a big funnel and get sucked into it, and they would like fold out in a particular structure. But these IDPs would have a flatter conformational landscape with little dips, that they would end up in and could easily be knocked out of, that sort of thing. And I may find a picture of those conformational landscapes and apply it here. Let's see, flatter local, okay, allowing IDPs to adopt a far wider spectrum of conformations, albeit less energetically stable ones, meaning that they can be knocked out more easily and flop around. So we should be developing more kind of a dynamic view of proteins in the cell, and more of a muddy liquid. I've always said the cell is a big mud puddle. More of a muddy liquid than a well-tuned machine with gears and levers.
Okay, so is it like mud that like you step in and you just kind of get mad because it got mud all over your pants and your shoes, but you can still walk on? Or is it the kind that you stick your foot in it and you get kind of stuck and you pull and finally you pull your socks out, or your sock feet out, and leave the boot in the mud?
And then you say, fuck it, and you just play in the mud like a child.
Yeah. So is there a difference there? I mean, how thick is it? Is what I keep asking myself. But it seems like it's thicker than just like plain water or, broth or something.
Yeah, it's like a really thick soup of kind of very hot fluid
Mhm.
that's flowing around very rapidly, and just happens to statistically create an environment where the cell stays alive and does what it does in that particular context. And that doesn't sound like a very good machine to me. So anyway,
Right. It certainly defies the conceptualization of a machine.
yep. And let's see, what else did I have? For many researchers, I'm trying to figure out what I highlighted here. Other functions, cell signaling can be accomplished by unfolded chains, sequence patterns, or isolated secondary structural motifs. Okay, you're on from a structural perspective.
One thing I noticed up here, from a structural perspective, IDPs pose an interesting challenge. The old idea that a protein binds to its substrate because their shapes match like a lock and key seem totally inadequate. As Chard playfully remarks, you might as well try to open the door with cooked spaghetti. So that's challenging another machinist, I guess, view of the cell, the lock and the key.
Yep. Let's see.
Kind of where do you want me to start?
Go ahead.
Where did you want me to start?
Oh, from the structural perspective, I guess. Okay. Please.
Okay, just keep
Yeah.
going. In the case of an IDP, the spaghetti uses the lock to mold itself into the shape of the key rather than forming the key beforehand. IDPs only acquire stable functional conformations when they bind to appropriate targets. Some, however, remain disordered even after binding, such as the signaling protein, I'm just going to say SIC1, which stays unfolded upon binding to CFC4. IDPs thus disprove the central dogma of structural biology as they empirically demonstrate that an ordered conformation is not, in fact, required for protein function. It appears, moreover, that this lack of a definite structure confers a considerable functional advantage on IDPs as it enables them to interact with a broad range of binding partners, including other proteins, membranes, nucleic acids, and various smaller molecules. By adopting different configurations, macromolecular aggregates containing IDPs also tend to display high degrees of conformational ambiguity, a phenomenon which has been termed fuzziness. That was a very popular term in physics for quite a while, I think.
Yep.
Still be. Here we are, once again, far away from how we think about structure and its relation to function in a machine. Indeed, the more we learn about the structure of proteins in their native state, the harder it is to uphold the mechanicist notion that protein complexes can be conceptualized and effectively, I can't believe I messed up that word, and effectively studied as molecular machines.
I think you messed it up twice. Immutable.
Oh, good.
So, catching up and reviewing for immutable, we found the methodology of studying proteins was to crystallize them very carefully in a laboratory and shoot X-rays in them and figure out the structure. And then when they started using NMR techniques to look at wild proteins, they found that they were more like a sort of a wax-like or semi-solid core with liquid around the edges, so that there was a lot of conformational changes going on. And then they found that that 25% of all proteins aren't even folded, that they interact as long strings. They don't even fold. And 50% of all signaling proteins have about 30 amino acids that are just sort of liquidly bouncing around and changing shape. So this allows proteins to fit into more of a, let's see, they described it
induced fit.
They described it as you might as well try to open the door with cooked spaghetti. The spaghetti uses the lock to mold itself into the shape of the key rather than forming the key beforehand.
Yeah, it's one of the things called it's one of the ideas called induced fit.
Yep.
And that is kind of both like in the case of proteins can, or the part of a protein that's binding can kind of wiggle its way around until it finds the right conformation to interact or to pass through in certain cases.
Yeah, I like this part. Proteins are really dense liquids or melted solids consisting of a near solid interior and a full liquid exterior. And I call a near solid interior a wax-like sort of thing. And let's see, what else do they say here? They call them intrinsically disordered proteins. They found a lot of them, and they have a function. Let's see. Okay.
That's actually not something I'm very familiar with. I need to read more about it though, because that's not something you'd cover in. That's not something you typically cover in general biochemistry. Like even
Yeah, Elberts wrote the molecular biology of the cell, that big Bible book. You know, the big heavy one. You drop it on your foot, you'll break your foot.
Yeah.
That every molecular biologist carries around, and he was one of the people that strongly promoted the machine-like idea of the cell.
Okay.
And he wrote most of the textbooks. So, anyway, it's your turn to read.
Let's see. Turning now to specificity, or are we there?
Yeah.
One sec. Turning now.
Problem.
Let me make this full screen. Gives it easier on my eyes. Turning now to specificity, it is also becoming apparent that the earlier generations of molecular biologists grossly overestimated the specificity of proteins. This is partially due to the fact that for most of the 20th century, methodological limitations require proteins to be studied in isolation from the cellular milieu in which they are embedded. Undoubtedly, one of the great appeals of the molecular machine concept is that it justifies ignoring this context, allowing researchers to focus their attention on the structure of the mechanical device and the mechanism of its operation. The problem is that when it comes to understanding what happens in a cell, context is everything. What a particular protein does, we now know, is largely defined by the environment it finds itself in, and the interactions it has with the molecules around it. Trying to acquire a complete picture of its behavior while overlooking these factors is a futile exercise. If I can pause for just a moment, haven't I been saying like the same thing for about a year on this fucking channel about that?
I can't remember. Were you?
That like proteins, essentially you have to consider them in like what environment you're finding them in. That the notion of innate function is kind of a vacuous idea.
Yeah, and it's stochastic and it's statistical.
Yeah.
And the only thing that matters is if the cell survives overall and performs the function it's supposed to function and doesn't decide to go all apoptosis on itself, kill itself. Cells commit suicide too.
I think we're a little further up, aren't we? Oh, no, we are.
No.
I've already indicated that in the case of IDPs, function is determined not by structure, but by context. This lesson can be generalized to all proteins. Functional promiscuity seems to be the rule rather than the exception for proteins. Even enzymes, which have traditionally been regarded as remarkably specific catalysts, exhibit varying degrees of catalytic promiscuity owing to the inherent conformational flexibility of their active sites, among other factors. A rather extreme case is the methanogen monooxygenase, which can hydroxylate 150 substrates in addition to methane. Not only are the enzymes catalytically promiscuous, but many of them also perform a range of non-catalytic functions, such as cell motility, membrane trafficking, chaperoning, activation and inhibition of metabolic pathways, and chromatin organization. This exciting discovery has come as such a surprise, given that it conflicts with the mechanistic expectation of specificity. That the phenomenon has been called moonlighting. A protein can have very different functions even if it does not undergo any post-translational modifications. Depending on where it is located in the cell, on the cell type in which it is expressed, and on the nature and the number of proteins it binds to, and on the amount of ligand, substrate, cofactor, or product available to it. As the multifunctional or moonlighting capacities of proteins are not coded in their genomic sequences, it is very difficult to predict them. Indeed, it is likely that many of the proteins that we think we know quite well actually perform additional functions that have not yet been experimentally identified.
Okay. So now the interesting thing about this is if one protein can have 150 different substrates that it attaches to, or if you can have 10 or 20 or 3 or even 4 different structures and hence different functions, you can see that when you think about the space of all proteins, when you think about neutral theory and mutations, you can see that a protein is more likely to find some function in a cell to fit into some pathway, hence evolution happens. Did I make that clear?
Yeah, I mean, it also kind of it defeats this argument of genetic entropy coming from these creationists.
Right.
Because that notion kind of relies on the fact that proteins have a designated and specified function given to them by their creator.
Very specific. Yeah.
Yeah. But it turns out to be the case that most proteins are quite flexible and they can perform a variety of different functions. And so, it'd be rather rare for you to mutate a protein in such a way that all of its functions that are critical for a cell to survive or for the protein to, kind of last and do its thing would be, done away with and just, melted out of existence.
Yeah, when you map out the protein space or the space of all possible amino acids for say 100 amino acid protein, you've got a space. And what this does, and then in that space you have these little points that are functional. And what this does is tell us that there are a lot more of those points. Douglas Axe famously came up with that. There is one in, what is it? The way he did it, it was 10 to the 73rd, I think was the middle estimate of the 10 to the 73rd of the 20 to the 150th. I don't know what that is. And do you want to do a quick calculation on what that is in base 10? So there are 20, if you have 150 amino acids, you have 20 to the 150th power different protein possible proteins, right? For 150 amino acids. Out of those, 10 to the 73rd have function. So we already have a tremendous number of, or I think it was 1 in 10 to the 70? Yeah, you divide that whole big number by 10 to the 73rd, and you end up with a tremendous amount of proteins that are still functional. But what he focused on is how 1 in 10 to the 73rd is functional, right? So he made it seem like it was really, really sparse. But then when you think about all the proteins when you think about the difference between 20 to the 150th and 10 to the 73rd, you have a hell of a lot of functional proteins left over. And that's the point that they miss in Axe's, I think it was in his 2003 paper. I'm not entirely sure. But anyway, so where was I? It is also worth mentioning that large-scale studies of protein-protein interactions have revealed that a typical number of interactors for a given protein is far greater than we previously assumed. And that's from 2005, which is later than Axe's work. This discovery becomes less surprising when we remember that the interior of a cell is a highly dynamic environment. Most proteins within it are rapidly moving about, continuously interacting with ever-changing partners. Associations among proteins tend to be stochastic and short-lived and are usually characterized by relatively low binding affinities. Clearly, the ambiguity, contingency, and context dependence of a protein-protein interactions are hard to reconcile with the exquisite specificity and tightly constrained operation that we would come to expect from a genuine molecular machine. Similarly, the transient nature of protein associations conflicts with the fixity and durability that we intuitively associate with arrangement of parts in a machine. And Scott, you're up next for a reading exercise.
Sure thing. Overall, the various findings I have discussed in this section regarding the structure and specificity of proteins, or lack thereof, are prompting a basic shift in how protein complexes are conceptualized. The potentially innumerable ways in which proteins can come together to form functional aggregates, the extraordinarily wide range of factors that can change their conformational state, and the dynamic and ephemeral nature of these associations has led some researchers to argue that many of the protein complexes found in the cell are better understood as pleomorphic ensembles than as molecular machines.
That's a
Drawing,
beautiful statement.
I'm sorry. What, sir?
That is a beautiful phrase.
Yeah.
Pleomorphic ensemble. Pleo means what? What's as a root, as a suffix?
Many.
So
Many morphic.
forms. Okay, that makes sense. Drawing especially on studies of protein complexes involved in intracellular signaling, these authors draw attention to the fact that these complexes are extremely diverse in size and composition and undergo numerous reversible post-translational modifications, e.g. phosphorylations, we'll go with that, in ways that drastically alter their conformation and activity. Receptor complexes, adhesion complexes, mRNA splicing complexes, trafficking intermediates, and many other kinds of protein associations do not exist in the cell as clearly delineated, structurally stable assemblies of fixed and highly coordinated subunits exhibiting a discrete number of conformations in accordance with the molecular machine model, but as fuzzy and transient ensembles with half-lives in the order of seconds or less, composed of weakly interacting and ever-changing subunits constantly flickering between alternative conformational states.
Yep. Okay.
Yeah. I mean, what that's telling you essentially is that cells and the way that proteins behave in cells, for the most part, they're kind of, they're changing their function kind of rapidly. They're they're adopting many conformations, and based upon what the cell needs, and what modifications are present on that protein, its function can change essentially like at a whim's notice, right? So it's far from this. So, and it seems to be that like it's far from this kind of defined structure that's like that mechanistically we're wanting to ascribe to proteins as being these machines that are stationary and kind of obsolete in terms of the function that they're described to have. Any comments on that one?
Go on, Immutable, as mayor.
Yeah. As Mayer put it, after considering the vast range of potential configurations that a single transmembrane receptor complex for platelet-derived growth factor, PDGF, can adopt, the activated receptor looks like a machine and more like a paleomorphic ensemble, or
Less
probability.
like a machine.
Yeah. Looks less like a machine and more like a paleomorphic ensemble or probability cloud of an almost infinite number of possible states, each of which may differ in its biological activity. Rejecting the molecular machine model has wide-range implications for how we study, represent, and explain protein associations and interactions. Importantly, it compels us to call into it compels us to call into question the widespread appeal to wiring diagrams and design charts, akin to those found in mechanical and electronic engineering, in schematic representations of metabolic, regulatory, and signaling pathways. An emblematic example is shown in Figure 2. Such engineering-based diagrams present compact summaries of protein-protein interactions, and by deliberately imitating the design of electronic circuit boards, they convey the impression and understanding of control. Visualizing cellular pathways in this way gives us confidence and emboldens us to speak optimistically about the current state of research in our particular fields. A good illustration in this last point can be found in a well-known paper by Hanna and Weinberg 2000, which made use of circuit-like representations of the cell to reflect on the state of cancer biology and define its agenda for the 21st century. Progress in dissecting signaling pathways has begun to lay out a circuitry that will likely mimic electronic integrated circuits in complexity and finesse, where transistors are replaced by proteins, example kinases and phosphatases, and the electrons by phosphates and lipids. few decades from now, having fully charted the wiring diagrams of every cellular signaling pathway, it will be possible to lay out the complete integrated circuit of the cell upon its current outline. We will then be able to apply the tools of mathematical modeling to explain how specific genetic lessons, or liaisons, serve to reprogram this integrated circuit in each of the constituent cell types so as to manifest cancer.
Okay, you guys want to discuss a little bit? I have a raccoon problem.
Oh, okay. Scott, you want to go first? Since I was.
I don't know. As far as commenting on what was just read,
Yeah, sure.
well, okay. For some reason, I can't get it to scroll up. Oh, but that's because I don't have control. It's like I have a piece over here and I can't control it. Let me just read this, and
Sure.
unless you have something to say about it.
No, I mean, I think what it's pointing out essentially is that that the mechanistic view is still very predominant in literature or even in academic research.
Right, like we're going to be able to reproduce all the functions of the cell and therefore predict, how a cancer is formed by which lesion appears in which part of the wiring.
Yeah, exactly.
Okay, so and I guess that's what we need to be able to do is do some sort of predictive modeling, but it can only take you so far.
Yeah, they had hope basically that we could outline it. We could figure out how cancer happens, and one of the reasons why cancer research has been so difficult for us to grasp is that it isn't quite that easy. It's more complicated, and I think we can go on here. The problem with these engineering-based descriptions and representations is that they do not accurately reflect biological reality. Did I interrupt you guys? I was feeding raccoons.
No, we're good.
Okay. For wiring diagrams such as Figure 2 to be useful as the wiring diagrams of electronic engineering, they need to assume a very high degree of specificity in molecular interactions and chemical conversions that are depicted as links in the circuits. And although this assumption of extreme specificity is perfectly consistent with the molecular machine model, it is not well supported empirically, as I have shown in this section. So that's an important little piece. Recent research strongly suggests that most protein-protein interactions are contingent and opportunistic and do not reflect a predetermined genetic design. It is important. That is a very interesting statement when you're arguing with the other side, right? It is important to realize that the wiring diagrams like Figure 2 illustrate only one of the many potentially innumerable ways in which a given set of proteins may interact with one another in the cell, depending on an eclectic range of factors and circumstances. Engineering-based representations of this sort are undoubtedly interesting to look at, but they are limited explanatory value. Now that is a pretty important concept right there. And I'm going to quit trying. What's that?
I said those are fighting words.
Those are fighting words, yeah. Limited explanatory value. And then the other part, let's see, what is it? The one of many potentially innumerable ways in which a given set of proteins may interact. So we could miss something by holding to the machine paradigm is kind of where he's the overall point of his paper entirely. All things considered, such representations probably do more harm than good, as they wrongly imply that the proteins featured in them reliably and predictably form the same exact network of interactions, which are envisaged again misleadingly as fixed solid-state molecular circuit boards. In doing so, these diagrams prevent us from appreciating the vast spectrum of alternative interaction networks that the same set of proteins can and do form in different cells, and even in the same cell at different times. Hence, what it is claimed, say that scaffold proteins are analogous to circuit boards, modular platforms that wire together components and direct flow of information, and can program complex signaling behavior, one should take such assertions and representations from which they derive with a generous dose of skepticism. And okay. You up, Immutable?
Yeah, I mean, I think before I do so, one question you might ask is, okay, like in terms of research, right? Should what we be focused on in that case kind of trying to understand or predict the dynamic nature of the cell now that we understand its mechanic and just not, frozen in a, in a little winter wonderland?
Yeah, how would you approach it? What I guess you almost end up with a statistical representation of what's going on in a system. But then again, we want to know the details of each interaction. But it requires, like, once we find a particular structural pathway and a structure and specificity, we have to keep looking for other contexts and other pathways that might be happening at the same time. In other words, we should not throw up our hands and say, oh, we have the circuit, now we're done.
Right. Yeah. There's other things it could be doing.
Yeah. I mean, I think what might be needed as well is to kind of amend this this machine view in terms of thinking about how do, okay, yes, there are transitions that proteins have that change their function, and yes, proteins are pleomorphic, so how do we now detect, like, when it would be appropriate to target, this type of protein, as performing, whatever activity is performing, right? I mean, I think that's kind of the relative train of thought.
Yeah, instead of one circuit diagram here, we may end up with many, many more, and it becomes a pretty big problem for biomedical engineering to realize that this may not be the last diagram you get from the same set of proteins.
I mean, the thing you have to now have to realize is that it's a dynamical system, so that this pathway, right? If that were to represent, say, a liver cell, right? That might represent it for some brief period in time, right? It might represent it for like 5 seconds or 20 minutes of the life of a liver cell, right? But you have to remember that liver cells are doing other things as well, right? So how does that pathway change with respect to time in terms of what the proteins are doing and in terms of what networks are interacting with what other networks, being a function of time?
Yep.
We're a little up from that, actually.
What's that?
We're on the little. There's a few little words before it goes into that.
Do you think those little words are important?
Maybe. The main differences between the two conceptions of protein complexes I have discussed in this section are summarized in
Table 2.
In Table 2.
Yeah, that's a good part of that section.
Yeah.
Yeah, and so it gives it gives actually an interesting kind of comparison.
The opposite of specificity is promiscuity.
Yep.
What are they saying about our proteins?
Our proteins are
saying that
Okay, we said it out loud.
Oh, I did? I thought I was thinking that. Okay, never mind.
No, I said it out loud. I think you did too.
I think I did too. But anyway. But
So in addition to grounding our theoretical understanding of the organization and constitution of the cell, the MCC has also influenced our view of the processes that take place inside it. To illustrate this, consider again the image of the cell as a highly intricate chemical factory, probably the most popular formulation of the MCC. The following extract from a recent textbook offers a typical articulation of this view. Speed.
Oh, hi.
Were you arm wrestling at the tune?
I was looking ahead, sort of, and trying to decide, okay, this is a new section, intracellular transport, power stroke or Brownian ratchet, which is kind of a new concept for us. But yeah, you can go ahead and read.
Cells act as chemical factories, taking in materials from the environment, processing them, and producing finished goods to be used for the cell's own maintenance and for that of the larger organism of which they may be part. In a complex cell, materials are taken in through specialized receptors, loading docks, processed by chemical reactions governed by a central information system, the front office, carried around to various locations, assembly lines. As the work progresses and finally sent back via those same receptors into the larger organism, the cell is a highly organized, busy place whose many different parts must work together to keep the whole functioning.
Okay.
Descriptions of this kind make clear that the cell must possess highly effective means of sorting, packaging, and transporting cargo to different destinations. The efficient delivery of molecular products to their intended cellular function is known as intracellular transport, and it is of vital importance for the cell's normal functioning. When it is disrupted, complications and pathologies inevitably ensue. Prima facie, the orderliness and efficacy that is imagined in the targeted mobilization of essential cargo inside a miniature factory contrasts rather starkly with the physical reality of the cellular milieu. At the microscopic scale, all entities exhibit constant stochastic movements as a consequence of thermal agitation. This phenomenon, traditionally referred to as Brownian motion, causes molecules in solution to perform random walks that result in diffusion. Although diffusion is a passive process, it nevertheless plays an indispensable role in the intracellular transport of small molecules, especially over short distances. For example, it serves as the primary basis for connectivity in signal transduction networks. However, diffusion becomes inefficient in the transportation of large vesicles and macromolecules. In such situations, the cell must make use of active and directional modes of transport, which are made possible by so-called motor proteins that carry cargo quickly and efficiently across cytoskeletal tracts. The discovery of motor proteins have long been assumed to have provided empirical support for the MCC.
Okay. So an interesting thing here is that in embryogenesis, in the development of an organism, there are microfilaments, microtubules, and lamins that exist between and just underneath the surface of cells on the inside and also on the outside of cells. And diffusion is very, very important, and that happens with Brownian motion. It's not precise, and you end up with all sorts of gradients that end up shaping the organism, that shape the development of the organism. And it isn't, there aren't, at this level on the outside of cells, there are not those little motor proteins walking along with cargo. Rather, there's a whole lot of Brownian motion and diffusion happening. And the degree to, I always describe it as between the cells, it's like you had cotton, packed in there. Packed to different amounts. So sometimes it's packed very loosely, sometimes there's no cotton at all, and it's going to have a great effect on the shape of the resulting organism from embryogenesis. So anyway, so who wants to read next?
I'll take a turn.
Okay.
Motor proteins convert chemical energy usually obtained by the hydrolysis of adenosine triphosphate into directional motion and the performance of work. That's adenosine, isn't it? I'm sorry.
Adenosine, that's fine.
There are many different kinds of motor protein, each of which performs a distinct motile function. Those that utilize the cytoskeleton for movement fall into two categories based on their binding partners. Actin motors, such as myosin, move along microfilaments through interaction with actin, whereas microtubule motors do something. Okay, we're done. As kinesin and dynein.
Kinesin and
I'm sorry, what?
kinesin and dynein.
Dynein. Okay, move along the microtubules through interaction with tubulin. These proteins also differ in the type of cargo they transport and then in their direction of travel. In accordance with the MCC, motor proteins are conceptualized as miniature versions of macroscopic motors. They are often described as tiny nanomachines that work in many ways, just like an automobile on the highway. As they consume fuel to power their motion and they move steadily in a directional manner at variable speeds along molecular highways, structural studies by X-ray crystallography have also been invoked to suggest that these proteins are reminiscent of large-scale machines. But how do motor proteins actually move? Are you talking, Speed? Because you're muted.
Speedy.
I'm
Hello.
sorry. Here is a nice little picture of these motor proteins and how they walk along and how they're depicted popularly in many YouTube videos. And this is just one idea. And he goes a little further, but I will let you continue.
Okay.
Okay.
The orthodox approach to explaining the directional movement of motor proteins is to appeal to the mechanical principles that govern the motion of man-made motors. In an internal combustion engine, for instance, the energy input delivered by the ignition of a combustible gas under pressure is tightly coupled to the performance of mechanical work, which manifests itself as power stroke. That results in the movement of the piston. In the same way, in motor-driven intracellular transport, it is argued that the energy input delivered by the hydrolysis of a high-energy compound like ATP induces a large amplitude conformational change in the motor protein, which generates a mechanical force, a power stroke that drives the molecule forward relative to a polymeric track. Appropriately, this is called the power stroke model, and it has dominated our theoretical understanding of how motor proteins work for decades. Sometimes the chemically induced conformational change in the motor protein that produces the power stroke is compared to the mechanical release of a viscoelastic spring, which thrusts the molecule forward. In the case of kinesin, which forms dimeric legs that alternatively attach to tubulin, the repetitive power strokes result in a hand-over-hand motion that makes the protein appear like it is walking along the microtubule. Figure 3 illustrates how the kinesin walk is commonly represented in the technical literature as well as in textbooks and other educational materials. Of course, animations such as the one shown in Figure 3 conform perfectly to what we would expect if the cell was indeed a machine, as they portray motor proteins as tiny robotic bipeds performing sequential cycles of precisely coordinated, mechanically powered movements along cytoskeletal tracts. However, upon closer inspection, it becomes apparent that these models of intracellular transport are fraught with problems. For a start, the plasticity, fluidity, and dynamicity that most proteins exhibit in vivo, which I discussed at length in the previous section, are difficult to reconcile with the rigidity, solidity, and stability that motor proteins would need to possess for them to move by power stroke mechanisms. Moreover, these models tend to overlook the fact that proteins operate in an environment that is drastically different from the macroscopic one in which we and our machines exist. Motor proteins, like all other molecules, are subject to constant thermal and quantum fluctuations that make carefully synchronized movements along a desired path challenging in the extreme. In fact, the energy of ATP hydrolysis responsible for generating the power strokes that allegedly propel motor proteins forward is only about an order of magnitude larger than the environmental stochastic forces that are permanently buffeting them. In such conditions, moving mechanically and deterministically is like trying to swim in molasses or walk in a hurricane. And there I see images of reporters standing, reporting about a hurricane, and then they get pushed pushed out of camera range because the winds are so strong.
Yeah, or a sign hits them in the head.
All
I
those things.
saw that in Florida. So anyway, so this model that we see, we have to consider that this may not actually be what's happening, because we've never sent a cameraman down here into the cell to take pictures of this particular little mechanism, right? So we have to consider that even though we love this model, this may have nothing to do with how it really works. Is kind of where he's going with this Brownian motion thing. Besides these general worries, a number of surprising empirical findings made possible by the use of novel methods have called into question the theoretical adequacy of power stroke models, such as the one illustrated in Figure 3. For example, although the hand-over-hand mechanism that underlies the walking motions attributed to kinesin is dependent on the protein's dimeric form, monomeric, dimeric means those two legs sticking out, two different, I think they're two of the same proteins that are attached together, so it's dimeric. Monomeric kinesin motors have been reported, which are equally capable of directional movement. More broadly, there's no obvious correlation between the amount of chemical energy a protein consumes and the distance it travels. Now, so they found empirically that we can't make it look like there's no correlation between how much energy is consumed, how far it gets. So that indicates a stochastic model. Single molecule measurements of myosin mediated transport have revealed that a single cycle of ATP hydrolysis can result in displacements of wildly variable lengths ranging from 5 to 30 nanometers. Additional studies of myosin movement indicate that the structural geometry of a protein's motor domain is not correlated with step size. These and other recent findings suggest that the structure of motor proteins may not be as crucial for their operation as one might have expected if these proteins were bona fide molecular machines performing precisely coordinated mechanically powered movements. Motor proteins also lack the functional specificity that is typically associated with machines, as many of them have been found to be, we got an illustration, involved in a number of additional non-motor cellular functions. Now, that's
he didn't.
What's that?
I went, oh, no, he didn't, because of that last statement there.
Yeah.
Non-motor cellular. In other words, because motor proteins are really important to a lot of factions of people, in that it's part of the idea of irreducible complexity. Oh, we've got these motor functions, we can't explain how they got there. But if those proteins have non-motor functions, then that they could have come from could have developed from a protein that not only had other functions but developed the new motor function.
Exactly. Irreducible complexity does not work in that case.
It almost makes it a very false notion of what happens in the cell.
Yep.
Well, and the thing is too, like even with like the most well understood motor proteins like ATP, like the F0 ATP synthase motor proteins, we understand most likely how they evolved, and that's by gene duplication. Because this thing is a heterodimer of the same subunits, meaning that it's just copying the same subunits to kind of solidify its structure for the kind of activity that protein is performing.
Yep.
And it would make sense that if molecular proteins are a ubiquitous phenomenon, they probably have a similar kind of evolutionary trajectory than the one that we understand already. But perhaps not.
Yeah, and dynein and kinesin are also involved in muscle formation, and I believe in movement in prokaryotes, basically with flagella, I think. And so these things have a number of functions, and you'll find there's a lot of correlation between these proteins when you look across a lot of different cell structure. And it's quite interesting that this is not irreducibly complex. So here's an illustration of the Brownian model which would fly in the face of that favorite animation of the Kinesin walking along, or the Dynean walking along. And I don't know how to make sense of this illustration yet.
Well, essentially what it's saying, if you scroll up a little bit.
Yes, sir.
So let's read go up so we can see a little more of it. Yeah.
Okay.
There we go. Schematic representation of Brownian ratchet model of intercellular transport. The motor protein attached to a cytoskeletal track is hypothesized to display two distinct potential energy landscapes depending on its conformational state. In the flip conformation, A and C, white ball, the energy landscape is flat, so the motor protein slides freely along the track, buffeted by stochastic fluctuations. So what that's saying essentially is that if you consider this stochastic model, right, or if you consider the conventional kind of model or the stochastic model, what it's saying is that the movement of the protein kind of depends on how many other things in the fluid that this protein is encountering. Imagine it like trying to walk on a very gusty day, right?
Walking in a hurricane.
Yeah, like if you're walking in a hurricane, you're not going to, no matter how much energy you burn, you're not going to be able to make it very far because there's just a lot of things that are in your way, and it's not very easy to progress. And so, the motion is kind of randomly determined.
Go ahead and read the rest of that.
Yeah, the motor protein slides freely along the track, buffeted by stochastic fluctuations. In the flop conformation, B and D, gray ball, the energy landscape has a sawtooth shape. So the motor protein drifts to the closest energy minimum, where it remains until it acquires the flip conformation. By periodically switching between these two conformations upon the repeated ATP hydrolysis, the motor protein is driven by thermal fluctuations to the right. So essentially what it's saying is that it's not necessarily like you're traveling through a flat landscape in this case. It's saying that you're walking through a landscape that has some areas that are more stable than others, right? So if you go back up, you can kind of see that in its depiction.
And you move forward.
Right. And when this protein reaches a state that's conformationally stable, or that's stable enough, right, it stops, and then it waits for another flop. So it waits till a point where it can move again in a way that it has a chance to get to the next conformationally low energy state.
Yep.
And it oscillates kind of between these two kind of behaviors of kind of fighting against this kind of high energy fluctuation environment, and changing its activity in terms of when it's moving and when it's resting at a conformation that's more stable for it.
Yeah.
So on this graphic here, the left to right, what would normally be the x-axis, that's basically moving along a microtubule or something.
Yeah.
Okay, and then what's the y direction? It says U. I'm not quite sure what the U.
The energy,
So,
the amount of energy.
It was like the energy landscape. So it's describing a landscape of energy in the protein where some conformations the protein hits are high, and some of the conformations are low.
Yeah,
So
when ATP hits, you enter the flip state, right?
Yeah.
And it flattens it. Basically, the ATP flattens it, and then it goes to the next ratchet. And because it is now in an energy well, it stops there, and that holds it from going backwards. So,
Right.
and then it runs into another flip state. And directionality, I'm not sure how he establishes that, but it has a direction.
It could go like, in the sense, it could, in terms of direction, it could go like 2 steps forward, 3 steps back, based on that Brownian motion. Is that what I'm seeing? That it could go other direction during that flip state.
You see how the energy well is sloped downward here in these ratchet in the flop state,
Right.
where it's sloped downward, so it falls forward basically into that one energy well, and that establishes direction. And then the ATP comes along, flattens it again, and rolls into the next one and the next one. So basically, the shape of the energy well is what establishes direction.
Okay.
So, okay. And an energy well, is like a lower energy state is more preferable for a molecule. Molecules like to be cool when they sleep, right?
Yeah.
Who
Basically.
doesn't?
Okay. So you want to go ahead and read? I think it's immutables up.
Sure. In light of these problems, in the last few years, a completely different account of motor-driven transport has started to receive widespread attention, known as the Brownian ratchet model. Although it is almost as old as the power stroke model, it has remained relatively unknown until fairly recently, presumably because it does not appeal to our mechanical intuitions in the way that the comfortingly familiar idea of a power stroke does. The basic contention of the Brownian ratchet model is that the directional motion of a motor protein is primarily driven by stochastic fluctuations and rectified or biased by chemical reactions, such as the hydrolysis of ATP. Empirical studies have established that motor proteins use the energy of ATP hydrolysis to flip-flop between two alternative conformations. What the Brownian ratchet model postulates is that the flip-flop conformations of a motor protein are characterized by different potential energy landscapes, as shown in Figure 4. In the flip conformation, A and C, white ball, the energy landscape has a flat shape. This means that the motor protein performs a random walk on its track, and as a result of thermal agitation, exhibiting equal probabilities of moving to the left or right of its initial position. In the flop conformation, B and D, gray ball, the energy landscape has a jagged, sawtooth shape. Consequently, random collisions jostle the motor protein overwhelmingly to the right, where it gets trapped in the nearest potential energy minimum trough. In this way, by stochastically switching between two different distinct conformational states, as a result of repeated cycles of ATP hydrolysis, the motor protein is able to harness the perturbations of Brownian motion to move in a specific direction. Along a cytoskeletal track. A fundamental difference between the power stroke model and the Brownian ratchet model is that the former takes chemical reactions, such as the hydrolysis of ATP, to be responsible for generating the mechanical forces that drive the motor protein forward, while the latter assumes that the chemical reactions serve to bias the existing Brownian motion in a particular direction. In other words, a power stroke motor moves despite stochastic fluctuations. A Brownian ratchet motor moves because of them.
Interesting.
In this respect, a very attractive feature of the Brownian ratchet model is that it explicitly factors in the counterintuitive physical conditions of the cellular milieu in its explanation of how proteins move directionally, instead of conventionally ignoring them or dismissing them as inconsequential.
So there's the example of a paradigm shift right there. Is explaining more than people are willing to see. The data is there, but under the mechanistic model or the whatever you want to call it, the MCC, nobody wants to deal with it, and so it doesn't get really factored in or emphasized. But a new model comes along, and suddenly it all receives the appropriate amount of weight.
Yeah, I mean it's nice to see these kind of arguments take place in scientific communities and paradigm shifts occur, because like he was saying earlier, right? These kind of paradigm shifts not only in terms of how we educate people, but it's also going to play a role in basic research and how we think in terms of developing drugs to combat disease and things of that nature.
Yep. So, Scott?
A further advantage of the Brownian ratchet model is that it is able to make sense of experimental findings that appear perplexing from the perspective of the power stroke model. The aforementioned reports of monomeric motors, the lack of correlation between the chemical energy used by the motor and the distance it travels, and the independence of step size from the structural geometry of the motor domain can all be straightforwardly accommodated within the Brownian ratchet model. In this model, structure and specificity do not play the same critical role in determining how the protein moves as they do in the MCC-derived power stroke model. Once it is adopted, the discovery that motor proteins are also involved in other cellular processes ceases to be baffling or surprising. Although the Brownian ratchet model is less intuitive and harder to initially grasp than the power stroke model, it is in many respects the simpler model of the two. As there is no specific reference to the topological or geometrical configuration of the motor protein, other than to its alternative energy profiles, which do have a structural basis, there is no need to speculate about how its various structural domains interact with one another in a perfectly synchronized fashion to generate motion. Similarly, because there is no crucial mechanical step, no power stroke, that can be identified as the specific moment at which chemical energy is transformed into work, it becomes unnecessary to invoke violent kicks, judo throws, or any other anthropomorphic actions to explain how motor proteins move directionally.
Ow.
Yeah. Ooh. But
Boy,
perhaps
he is punches. Fucking hell.
this guy's ready to go to bring it on.
Yeah, we're not picking on creationists here.
No, not at all. Not at all.
No, but I mean, it is kind of interesting how feisty this author is getting, getting riled up with, the MCC model.
Yep. I wonder if he would like to come and talk to us.
I think that might be a good idea. I'd also like to see, because I have the paper pulled up. I'd like to see if he's published any further literature on this too.
Oh, he has many.
He published a lot.
In fact, he has an 87-page paper on normativity in biology, which Danny was excited about. So that's something we'll probably take a look at some point. But 87 pages, it'll take us the rest of our lives to get through that, right?
Come on, if we just make it through one page a day, it'll take us like a third of a year.
That could be the rest of my life.
You're so optimistic, Immutable. It's just cute. It's just cute.
I know. Aren't I just a little sweetheart that way?
Keep in mind that you live in a place where there are hurricanes and crazy Republicans, drug lords, and alligators.
And pythons.
Oh, God, no wonder. This guy is a philosophy of biology kind of guy. Oh man. All right.
Yeah, he is. Yeah. I will show the picture of, well, in fact, I thought I did show the picture of his page where he has all his publications. He has quite a bit. And he looks like a rather young man. So he's going to be around for a while. Okay, so who wants to continue? I guess I could for a while, but I hate to read. But perhaps the greatest theoretical value of the Brownian ratchet model is that it elegantly demonstrates how stochasticity can be put to good use in the cell. And the cell is a place where you're going to find a hell of a lot of stochasticity. Whereas the power stroke, I made that part up. Whereas the power stroke model considers motor-driven transport to be an example of what Schrodinger called the order from order principle, given that complexity of the mechanism it postulates is assumed to derive from a pre-existing genetic design. The Brownian ratchet model regards it instead as an instance of order from disorder. Now that is interesting, called, okay, I want to highlight that, order from disorder principle, which Schrodinger claimed, incorrectly as it turns out, plays no role in biology. This is because it shows how the coupling of two random or disordered processes, namely Brownian motion and the binding of ATP, can result in a non-random or ordered outcome, directional movement. In this way, by providing a non-deterministic, design-free conceptualization of intercellular, and we have to go to another page. I'm working on it. Transport, the Brownian ratchet model strikingly illustrates how order can be generated out of chaos. Nice. Could we quote mine this for debates?
Well, I don't think I quote mining if you actually quote relevant parts.
Yeah, we could actually quote it, like in full.
Yeah, that's a thing. I think. I hear.
You know, it's kind of nice when people like quote something in its full context instead of, obviously parse out a seemingly interesting part of a paper that, creationists tend to ignore the rest of it when it disagrees with them.
Too bad there isn't a YouTube channel, Speed of Sound of Gravity, which goes and reads the entire paper.
I know. This needs to be a more common occurrence.
Yeah.
I was thinking of like you ought to create a playlist that says paperwork or whatever you want to call it, paper trails.
Paper trails, yeah.
Paper trails, there we go.
You just stick it in that playlist.
Yep. Okay, so mutable. I want to go ahead and we have a column and a half, a column and a 3/4 to read before 9 o'clock.
Oh Jesus! Well, wait. How much further do we have with this thing?
Just a column and 3/4, and then we get to the next section over here. I think right there, cellular behavior.
Ah, nice.
So we're almost done with 2 sections today. So okay.
Just wait till we hit some of these origin of life papers, which are like 50 pages long. And
I know.
Fucking God.
You're up.
It is undeniable that the growing use of single molecule technology in experimental studies of motor proteins has greatly contributed to raising the profile of the Brownian ratchet model. However, there's still no consensus as to whether motor-driven transport is best understood in terms of the increasingly popular Brownian ratchet model or the more traditional power stroke model. Advocates of the former maintain that power strokes are irrelevant in determining the directionality, stepping force, and optimal efficiency of motor proteins, while supporters of the latter insist that motor proteins would not be as fast and powerful as we know them to be if they operated by a Brownian ratchet mechanism. Some claim that the two models sit at opposite ends of a continuum within which most motor proteins actually operate. For example, Oster and Wang, 2003, page 208, assert that there are only a few motors that can be regarded as being pure power stroke motors or pure ratchets. Most protein motors employ a combination of the two strategies. If this is true, then the two models are not necessarily mutually exclusive. At the present, it is not yet possible to settle this theoretical dispute. The two models, we should not forget, constitute different attempts to interpret the same empirical data. They are
Second.
Yeah.
I'm sorry.
No, they are extrapolations from experimental studies. No one has actually seen a kinesin literally walking along a microtubule.
Oh, we haven't observed it?
Well, I've never seen it like up close and said like, "Hi, how you doing? You want to go on a date?" type of thing.
Yeah. You mean we haven't observed it? Have you ever heard that before? Okay,
Oh,
go ahead.
Maybe this is like Maddox's long lost father.
Okay.
Nevertheless, what can be asserted with a reasonable degree of certainty is that the engineering-based power stroke model, at least when conceptualized in analogy with the power stroke mechanism of an internal combustion engine, if not irrevertibly flawed, at best offers only an extremely idealized interpretation of motor-driven transport as it occurs in the cell.
What is it?
Go ahead.
Okay.
You want me to continue?
I'll do it.
Okay.
Still, even if the Brownian ratchet model becomes unanimously accepted as the preferred explanation of how motor proteins work, it could be argued that recognizing that the cell contains a real motors is already one concession too many to the MCC. This, however, does not follow. Molecular motors, if that is how one is to refer to motor proteins, are not miniature versions of macroscopic motors. In fact, they differ from macroscopic motors in almost every important respect.
Oh,
For one thing, they lack rotors, armatures, and all the other trappings of conventional motors. They are made of soft, flexible materials which exhibit high degrees of freedom, unlike the hard levers, cranks, and hooks that make up most mechanical devices. Moreover, due to their minuscule size, the influence of gravity and inertia on their operation is insignificant compared to that of the raging Brownian storm that permanently engulfs them. This turbulence, combined with the high vicious drag of their fluid environment, makes the long-range transmission of precise mechanical forces physically impossible. In addition, as I have already indicated, whereas in man-made motors energy is used to drive motion, in molecular motors energy is used to restrain motion. The former move directionally by overcoming stochastic perturbations. The latter do so by exploiting them. A further difference.
sorry.
Let's see, where did I go?
A further difference.
Yeah, a further difference is that molecular motors convert chemical energy directly into work without using heat or electrical energy as intermediates, which is why their efficiency is much higher than that of macroscopic motors. Overall, because of all these crucial differences, although it may seem tempting to draw analogies between molecular motors and macroscopic ones, we should keep firmly in mind that in answering fundamental questions regarding problems associated with friction, wear, transmission, efficiency, fuel, motion, and work, such such facile comparisons often serve to cloud rather than simplify issues.
Now, the interesting thing there is that it seems that life has basically captured the storm, the Brownian storm, raging Brownian storm,
Yep.
and used it to its advantage and ended up with a more efficient mechanism than we would get from a mechanical motor, which is interesting. And then he has his table up here, but I think we start reading here and we're almost done. So.
Ultimately, the issue boils down to how we choose to define the term motor. We tend to assume that motors constitute a class of machine, hence the objection I have just considered. But perhaps we should reverse this relation and consider machines to be a class of motor. After all, a motor can be defined very generally as an entity that imparts motion. In fact, this is the first definition of motor that is listed in the Oxford English Dictionary. Given that machines are not the only entities capable of imparting motion, it follows that not all motors are machines. As I noted in the introduction, the word machine tends to carry a number of additional connotations, such as a pre-existing design, a tightly constrained operation, and a deterministic outcome. It is therefore possible to conclude that motor proteins are indeed genuine motors, even though they are not machines. It is interesting to observe that some authors are starting to display an awareness of this important distinction, as the following passage suggests. Because they operate inside of a cell, molecular motors are tiny and operate on a physical scale that makes them very different from the man-made, macroscopic objects we normally imagine when we hear the word machine. Further, their size and soft structure allows them to be much more dynamic and robust than artificial machines. They work needing very little input, as energy levels not far from average thermal energy are sufficient for a given task. This property, too, contradicts with artificial machines, which work much more rapidly, accurately, and deterministically, but with higher energy demands and less adaptability. The main differences between the two conceptions of motor-driven intracellular transport I have discussed in this section are summarized in Table 3.
Okay, so
Jesus, hell, that was a lot.
yeah, and that brings us to 5, which is a good breaking point for this video. But I want to point out that Matt Ox's criticism yesterday involved the definition of a machine, and they are defining a motor as something different. So not all motors are machines. So if the cell if the mechanism of transport is a motor, it is still not a machine. And he uses this. We can stop this for the moment and share screen. Chrome tab. And right here. So he basically, and I wish I could make this bigger, and maybe I can by doing this. Mini player? No, we want that. No, we don't want mini player. Damn mini player. Stop it. Stop it, mini player. Oh, I just screwed up, didn't I?
Let's go back to your history.
Let's see. Where did it go?
Well, you lost the player, so I think it's just taking you back to your home position. But I think if you go over to the history tab, you'll be able to find it.
Right here.
You know, I do wonder how Otangelo is willing to, what Otangelo will say when the series is done, given that this is kind of one of his sticking points.
Yep.
Yeah, I already told him in one of the live chats that his machine model is rather passé, and he needs to catch up on the literature. But I was exaggerating a bit there because I just tend to think in terms of how fruitful is a particular paradigm for inquiry and in getting experimental results. And it's just that it seems that the machine model is less and less useful for that. And when you go into this pleomorphic model, I guess you would call it, then you tend to account for more phenomenon within the cell than you do mechanistic model.
Yeah.
And so I would say that's partially what we need to do if we want to understand kind of the, some of the details that are clouded, that appear kind of fuzzy when we look at things from a mechanistic model.
So a machine or mechanical device is a mechanical structure that uses power to apply forces and control movement to perform an intended action. They can also include computers and sensors that monitor performance and plan movement, often called mechanical systems. So he's in this, he is pushing back against that idea that it is that kind of a thing where it applies this power stroke and rather it relies on a more stochastic, energy-rich and fluid environment to get the job done, which is...
Yeah, Nicholson also lists the criteria for what you have to have a machine at the beginning of the paper. So, he sets up his own working definition of machine. I don't know if we compared it to the Wikipedia model of the machine, because that's what that is.
Yep.
Then I don't know if there would be any real difference, but
I'd love to see if this guy would be willing to chat about this at more detail.
He might come on, and I will try to get a hold of him and send him a link to our papers and see if he has any pushback or we got anything wrong or misread him.
Yeah, I would be. Well, I guess I shouldn't be embarrassed because I'm just a juvenile when it comes to all this. But yeah, I wonder if any of the conclusions or statements I've made are just like totally idiotic compared to what he had in mind.
I think you are quickly graduating out of the juvenile class, Scott.
Well, I'm about time.
If he does agree, I do have questions in terms of like the practicality of this idea. I mean, because, okay, yes, I can acknowledge that perhaps there needs to be a paradigm shift, right? But, in terms of like the educational sphere or in terms of how this shapes research, right? How does it influence, the trajectory of research and the types of experiments that we as, scientists are going to design and, develop to treat diseases and stuff like that? Right?
Yeah.
How do you,
Be interesting to find out how he's been received. This is from 2018, so it's been on the revised, accepted on June 2019. So it's about a year old, or less than a year old. Over a year. Yeah. What's that?
How many citations?
39.
39 citations. Okay. Yeah, we could look at a few of those and see and check on how it's been received. And it would be interesting to see whether he's gotten a lot of pushback or hate mail or or love letters. Okay, so let's wrap it up for this video. What do you think?